It's simple structure. Archaebacteria structure also very simple. If we compare the other bacteria structure, Archaebacteria structure is very simple. And peptidoglycan, one of the important point is peptidoglycan is absent. This peptidoglycan, unlike other bacteria, few bacteria, cell wall possesses peptidoglycan or uramic acid is there. But here, peptidoglycan is absent in the cell wall. Cellulosic or carbohydrates are present. And because of the cell wall nature, it it can tolerate the very harsh conditions or extreme conditions. Harsh and extreme condition means what? Salt conditions, and also hot springs. Hot springs means the temperature is more than 50 degree temperature. In hot springs, the bacteria can survive. And marsh type of soils, marsh type of in this nature also the bacteria can survive. In the animal rumen gut system, the gut system, the this type of the archaebacteria can survive. Some of the cow, buffalo in the intestine, archaebacteria, methanogenetic archaebacteria is present. Actually, in acidic side, if it is present, acidophilic bacteria. Okay. And in salt conditions, this bacteria is present because halophilic bacteria. So, depends upon the surviving present of the nature, halophilic nature, halophilic bacteria, halophilic, halophilic bacteria, acidophilic bacteria, acido, acidophilic bacteria. So, depend upon present its nature, wherever it is grown. Actually, these are the extreme conditions. Hot springs means the temperature is more than 50 degrees or near to 100 degrees. Generally, bacteria, our true bacteria surviving temperature is 37 temperature, between the 30 to 40 degrees temperature, surviving of that temperature. But more than 40 degree temperature, bacteria are not surviving, unable to survive more than 40. But Archaebacteria can survive. Archaebacteria survive because of its cellular nature. It's very primitive, having its very primitive character. It can survive in marshy soils also, in hot foods also, in soil condition also, in animal intestine, rumen also, bacteria can survive. Bacteria can survive in animal intestine also. So, the antibacteria, because of process, we know that in the movement called as a methanogenic bacteria. So many times exams asking methanogenic, methanogenic bacteria, So methanococcus bacteria, methanogenic bacteria. With this bacteria in, in the animal intestine, that present this bacteria. After the in animal dung, we found this animal dung. Animal dung. हिंदी में क्या बोलते हैं गोबर बोलते हैं आग पे डाल गोबर बोलो. So that bacteria is present. With this animal dung, we can create the biogas. We getting the natural gas, biogas. So the biogas production with this archaebacteria. So not unlike our other type of the bacteria, it is a very complex in nature. Human bacteria are very complex. This is a very simple structure, simple bacteria. Okay, and if you study gene level, if you study internally gene level, up to DNA structure gene level, these bacteria, archaebacteria are possess introns. Introns are present in bacteria. Introns are present. Introns are the coding sites. Coding non-coding sites are there. Exons and introns, the DNA. So take the DNA, the structure of the DNA. There are some sites you call as a, some of the sites you call as a exons. Some of the sites you call as a intron. Including human beings also having exons and introns are there. Exons are actually coding sites. These are called as a coding sites to code the genetic character. The introns are non-coding sites after the splicing process. There is a process in the nucleus or in the DNA takes place. In the splicing, these introns are removed. These introns are removed. These are the non-coding sites. in the DNA of organisms. Here, in Archaebacteria, these introns sites also found. Introns are present in Archaebacteria. Unlike our bacteria, in eubacteria, these introns are absent. Introns are absent in bacteria, DNA of the eubacteria, but Archaebacteria, introns are present. Introns are present. So, molecular level observation that these Archaebacteria also possess the introns. Okay, this is information about the Archaebacteria, and in examination, it asking the differences between the Archaebacteria, eubacteria. You can write like the this is a very primitive one. This is some modern type of the bacteria. Eubacteria is a modern. The structure is simple. Archaebacteria structure is simple. Eubacteria is a complex type of the structure. And peptidoglycan is absent. There eubacteria peptidoglycan is the present. Like that. And it can survive harsh conditions. But eubacteria are not survive in harsh conditions. In harsh conditions, these eubacteria are not tolerated. The conditions are not tolerated by eubacteria. And the introns are also present in the archaebacteria. Here the introns are absent. In the DNA level, introns are absent. This is all about the archaebacteria character. I hope you understood this. Archaebacteria in short question in competitive exams also. All competitive exams also, archaebacteria is very important also. Asking so many questions like extreme level bacteria, which is the extreme level bacteria, is the archaebacteria. Which bacteria grown in salt condition? Archaebacteria. In which bacteria found in hot springs? Thermophilic bacteria. That is the archaebacteria. Thermo means in hot temperature also. Which bacteria means archaebacteria. Which bacteria giving the biogas? Biogas. We use it in our home and some type of the gas. That is the liquid petroleum gas, LPG gas. And in that gas, we found the butane. RN protein is present in that gas. Okay, so many times asking exams. What type of the gas is present? Liquid gas is present in that our cylinders, our home using cylinders. That is the liquid petroleum gas. But in the biogas is a methane. The gas is methane. The methane gas because of methanogenetic bacteria. The methanogenetic bacteria is belongs to a group of archaebacteria. Okay. So this is the information about the archaebacteria. Okay.